Hey science lovers, welcome back to my channel. Imagine a virus so intelligent that it can decide whether to kill its host or quietly hide inside it, waiting for the perfect moment to strike. Sounds like something out of a science fiction movie, right? Well, that's exactly what the Lambda Phage does. This tiny but powerful virus infects E. coli, and its ability to switch between two life cycles, one where it rapidly multiplies and destroys the bacteria, and another where it silently integrates into the bacterial genome and makes it one of the most studied phages in the microbiology. Today, we are going to explore the structure, genome, infection process, and life cycle of Lambda Phage in detail. By the end of this lecture, you will have the clear understanding how this virus makes so life and death decisions and why scientists use it as a model in genetic research. So, what exactly bacteriophages are? Bacteriophages, commonly called as phages, are viruses that infect and replicate inside the bacteria. Unlike viruses that infect humans or animals, phages are highly specific to bacteria. Each phage type is tailored to infect only certain bacterial species by recognizing specific receptor proteins on their surface. To give you an idea of their abundance, there are an estimated 10 to the power 31 bacteriophages on the earth. That's more than the number of stars in the entire universe. You can find them everywhere in soil, water, the human gut, or even in the extreme environments like hydrothermal vents or Arctic ice. In the ocean alone, phages destroy nearly 40% of bacterial cells every day, playing a major role in maintaining the microbial balance of ecosystems. But why should we study phages? Their significance extends beyond the ecology. They are the powerful tools in the molecular biology and medicine. Scientists have used phages in the genetic engineering, gene cloning, and even as vehicles for gene therapy. One of the biggest emerging applications of phages is in phage therapy, where bacteriophages are used to treat bacterial infections, especially those resistant to antibiotics. Since antibiotic resistance is a growing crisis, phage therapy is being explored as a potential alternative to fight superbugs like MRSA and drug resistant tuberculosis. Phages can be classified based on their life cycle. There are two main types. First one is lytic phages, also known as virulent phages. This immediately takes over the bacterial cells, use its machinery to replicate, and then burst the cell open, releasing the new phages. A classic example is T4 phage, which infects E. coli and destroys it within minutes. Second one is temperate phages. Unlike lytic phages, temperate phages like Lambda Phage can choose between immediate destruction or integrating their DNA into the bacterial genome. This allows the phage to stay dormant and reactive later under the stress conditions. Now, now that we understand what bacteriophages are, let's zoom in on Lambda Phage and analyze its structure in detail. The lambda phage is a complex virus composed of three major parts: the head or capsid, tail, and tail fibers. Each of these structures play a crucial role in ensuring the virus successfully infects the E. coli and replicates inside it. First, let's talk about the head, also known as capsid. It is icosahedral, meaning it has 20 triangular faces, which is a common structural feature of many viruses. The head is made up of coat proteins, GPE proteins, and serve as protective cell for the double-stranded DNA genome, which is about 48.5 kilobases long. This DNA contains all the genetic instructions required for viral replication and host manipulation. Next, we have the tail. Unlike some other phages like T4, which have contractile tails that shrink to inject the DNA, Lambda phage has a non-contractile, flexible tail. This tail acts as a tube through which the viral DNA is delivered into the host cell. The tail is primarily composed of GPV proteins, which form a stable channel. At the very end of the tail, we have the tail fibers. These fibers are responsible for binding to the host cell surface. Specifically, lambda phage recognizes a protein called LAMB or LAM protein, which is found on the outer membrane of E. coli. This is what gives lambda phage its host specificity. It cannot infect just any bacteria. It must find a cell that expresses LAM protein. The key protein responsible for this binding is called GPJ protein, which acts like a molecular key that fits into the bacterial lock. So, to summarize, the lambda phage is a highly specialized biological machine consisting of a protective capsid, a tail for DNA delivery, and a tail fibers that ensure specific attachment to the E. coli. This entire structure is designed to efficiently locate, attach to, and infect its bacterial host, setting the stage for viral replication and multiplication. Now that we have understood the structure of lambda phage, let's dive into one of the most fascinating aspects of this virus, its genome organization. The Lambda phage genome is a well-organized genetic blueprint designed for efficient infection, replication, and survival inside the bacterial host. Lambda phage carries double-stranded DNA and its genome is approximately 48.5 kilobases long. Interestingly, the genome exists in two different forms. Inside the phage particle, it's linear, you can see in the image, with special sticky ends called cos sites. Once inside the host cell, the cos sites allow the DNA to circularize and making it ready for replication or integration. These cos sites are crucial because they ensure that the lambda phage genome can function properly within the bacterial cell. Without this circularization, the DNA would be unstable and prone to degradation. Now let's look at the key genes that control the lambda phage's life cycle. The genome is divided into four main regions. First one is regulatory genes. So these genes determine whether the phage enters the lytic or lysogenic cycle. The most important players here are CI, lambda repressor. This gene is responsible for maintaining lysogeny. It suppresses the expression of lytic genes, allowing the phage to integrate and stay dormant inside the host genome. Now, CRO means control of repressor and other genes. This gene is the opposite of CI. If CRO dominates, the phage switches to lytic cycle, leading to viral replication and bacterial destruction. N and Q. These are transcription regulators that allow the phage to express early and late genes needed for replications. Now the second one is structural genes. These genes encode the proteins required to assemble new virus particles. They include GPE or capsid protein forms the icosahedral head. GPV or tail protein forms the long tail tube. GPJ or tail fiber protein allows attachment to E. coli receptors. The last one is integration and excision genes. The lambda phage can integrate into the bacterial genome and become a prophage. This is done by the INT or integrase gene. INT gene, which inserts the phage DNA into the bacterial chromosome. When condition changes, the phage can excise itself using the XIS or exionase gene and reactive the lytic cycle. Now that we understand the structure and genome of the lambda phage, let's discuss how it actually infects a bacterial cell. Infection starts with the attachment, which is first and most crucial step in the phage-host interactions. Unlike some phages that randomly collide with bacteria, lambda phage is highly specific. It only infects E. coli strains that express a particular membrane protein called LAM protein. This protein normally helps bacteria take in maltose, but lambda phage exploits it as a receptor to gain the entry. The tail fibers of lambda phage contain a protein called GPJ, which acts like a molecular key recognizing and binding specifically to the LAM protein. This ensures that lambda phage only attaches to suitable bacterial hosts, avoiding non-compatible bacteria. Now, once bound, the phage does something unique. It does not use a contractile tail like T4 phage. Instead, it undergoes a conformational change you can see in the image, allowing its linear DNA to pass through the tail tube into the host cell. This injection happens passively using the bacterial proton motif force to assist the DNA transfer. However, the DNA enters the cell in a linear form which is unstable. To stabilize it, the phage uses a special trick. Its cos sites, which are complementary sequences at both DNA ends, allow the genome to circularize. This is essential for replication and integration into the host genome. At this stage, the phage must decide between two fates: lytic cycle, activate replication and cell lysis, or lysogenic cycle, dormancy inside the bacterial genome. Let's now explore how lambda phage makes these life or death decisions. Now comes on one of the most fascinating aspects of lambda phage biology, the decision between lysis and lysogeny. This decision is not random, it's a highly regulated genetic switch influenced by conditions inside the bacterial cell. If the host cell is healthy and growing, the phage prefers the lytic cycle. This means it immediately replicates, produces new virus particles, and eventually bursts the cell open, releasing about 100 new phages to infect other bacteria. On the other hand, if the bacterial cell is under stress, like nutrient starvation or DNA damage, the phage takes a safer route. Instead of killing the host, it integrates into the bacterial genome and remains dormant as a prophage. This is called as the lysogenic cycle, and it allows the phage to hide inside the bacterial DNA until the conditions improve. The decision is controlled by a genetic battle between two key regulatory proteins. CI or lambda repressor. If this dominates, the phage remains dormant. CRO or control of repressor. If this dominates, the phage enters lysis and kills the host. So this balance determines whether lambda phage acts as a silent passenger or an active killer. But what happens when the lysogenic phage decide to wake up? Let's explore how prophages excise and enter the lytic cycle. Lysogeny is not permanent. Under specific stress condition, lambda phage can wake up and re-enter the lytic cycle. But what triggers this switch? One of the major triggers is DNA damage, which can be caused by environmental factors like UV radiation, antibiotics, or chemical stress. When bacterial DNA is damaged, A bacterial SOS responses is activated, which also signals to the phage that conditions are no longer safe. A protein called RECA detects the DNA damage and triggers the degradation of CI repressor. This means lytic genes previously suppressed are now activated. Two key phage genes, XIS or xCionase and INT or integrase, work together to cut the phage DNA out of the bacterial genome. Once excised, the viral DNA circularizes and the phage reactivates the lytic cycle, leading to host cell destruction. So we have covered a lot today. To summarize, Lambda phage is an incredibly well-studied temperate phage that infects the E. coli. It has a structural genome that enables efficient decision making between lysis and lysogeny. Its ability to switch between dormant and active states has made it a powerful tool in molecular biology. I hope you found this deep dive into Lambda phage fascinating. If you enjoyed this, make sure to like, subscribe, and leave a comment on what topic you would like the next. See you in the next video. Thank you.